Hello everyone, and welcome to episode 80, which will be an exciting episode that'll continue on with this multi-part series on animal physiology. Today we'll be exploring the dynamics of animal gas exchange, or animal respiration. Now this episode is the second in this second series on animal physiology, and this makes it analogous to the animal hydration episode in the previous series on animal physiology. And I did that on purpose. Both water and oxygen are critically necessary for animal life. Without either one of these crucial resources, the animal will quickly die. End of story. Some animals can endure droughts or long periods of time without drinking, like camels, just as other animals are specialized to endure long periods without breathing, like sperm whales. But for all animals given enough time, without water or without oxygen, death is inevitable. The big difference here is that while an animal, perhaps larger animals, can survive for at least a few days without water before they die of dehydration, most animals can only survive for a few minutes without oxygen, maybe even a few seconds, depending on their size. Insects, for example, will die if they go without oxygen for just a few seconds. Humans will suffocate in a few minutes. The point I'm trying to illustrate here is that oxygen is an immediate, critical need for an animal organism, which largely gets used immediately for cellular respiration and oxidative phosphorylation to produce chemical energy. More specifically, the ability to inhale oxygen and disperse it throughout the body, and to extract waste carbon dioxide and exhale it from the body, is of paramount importance to the animal. Without oxygen, the animal suffocates from a cellular level. Without waste CO2 being released, the animal can experience CO2 poisoning and die. So, without sugarcoating anything, gas exchange in animals is an absolutely critical aspect of their existence. And how exactly this all works on a cellular level in response to these immediate needs, that'll be what I'll discuss today. And in addition to respiration and gas exchange, I'll also be exploring the animal's circulatory system. And this is because gas exchange in larger animals requires the circulatory system, as larger animals require deep vascular perforation within their tissues to maximize the diffusion of both nutrients from food and the gases used and created through respiration. Okay, so I'll begin at a natural starting point, when oxygen is first consumed by the animal's body. This is called ventilation, which is a very general term to mean the stage where gases are exchanged across an animal's organ. So you have oxygen that's coming into the body of a fish through the gills, for example. That's ventilation. Carbon dioxide leaving the mammal body when it's diffused out of the blood through the alveoli into the lungs where it then gets exhaled, that is also ventilation. On a chemical level, this gas exchange occurs at the surface of the organ, at the interface between the chaotic outside environment and the organized chemical superstructure that is the animal's body. As this gas is absorbed into the bloodstream, it'll get circulated around and taken where it needs to go. Oxygen is delivered through the blood to tissues that need it to produce energy, and waste carbon dioxide moves through the blood to the lungs to be removed. The critical step is cellular respiration, where molecules of these gases are used by individual cells to stay alive and produce the energy they need to perform various cellular subfunctions, and the waste carbon dioxide they produce needs to be expelled, removed from the body. This aerobic cellular respiration is an ancient chemical process that's so efficient and so powerful that the animal's macroscopic bodies have literally evolved around it so as to facilitate and support this process of oxygen inhalation, oxidative phosphorylation to produce ATP that powers all of the processes of their large, complex bodies, and then exhalation of the waste CO2 that is a product of respiration and all sorts of other metabolic processes. To begin, I want to talk about the beginning and ending stages of the process of respiration, where ventilation occurs as gases are exchanged across membranes. To explain how this works, I first need to touch on Daltons Law, which states that a mixture of gases applies a pressure within its volume, and that the component gases within this mixture can have their partial pressures calculated. Now, the partial pressure is just the portion of the total pressure that an individual gas is responsible for. All of the partial pressures of all the individual gases in a mixture added up will equal the total pressure. In the real world, this manifests as the atmospheric pressure. The atmospheric pressure is measured in millimeters of mercury, or the pressure that's applied under the mass of a column of mercury X millimeters tall under standard temperature and pressure at sea level. The atmospheric pressure is 760 millimeters of mercury, and oxygen, composing just 21% of the atmosphere, and thus 21% of that total atmospheric pressure, thus has a partial pressure of 160 millimeters of mercury. Now, it's important to understand partial pressure, because gases will tend to flow in the direction of their partial pressure gradient, just like how solutes will tend to flow in the direction of their concentration gradients. Second, I need to explain Fick's law, which calculates the rate of gas diffusion according to K, the diffusion constant. A, the area for gas exchange, P2 minus P1, which are the differences in partial pressure, and the distance of the barrier to diffusion, or D. So this creates a clean little equation that goes K times A times the result of P2 minus P1, all divided by D, which can then be applied to virtually everything, including biological systems. So for example, in the human lung, the epithelium of the alveoli have a huge surface area, so A is large. The thickness of the membrane of these alveoli cells is measured in decimals of a micrometer, so D is small. Now, oxygen diffusion into the animal is maintained by having a large difference between P2 and P1, which promotes gas exchange exactly like how a larger difference in solute concentration will create a stronger gradient. So now all of these numbers, multiplied by K, which represents the solubility of oxygen gas at a human habitable temperature, this can tell us our optimal rate of oxygen diffusion into our bodies. Furthermore, there are aspects of the solution that influence how gases are absorbed and diffused. In cases where the natural physics of gas absorption aren't really favorable for life, life can evolve ways around it. For example, oxygen naturally dissolves pretty poorly in water. Something like 0.003 milliliters of oxygen will dissolve per 100 milliliters of water, per each millimeter of mercury in pressure. Now, this natural rate of absorption is, on its own, not able to sustain life. So animals evolved enzymes like hemoglobin that can bind to this oxygen and facilitate its dissolution and diffusion through the blood. In the context of the ocean and the atmosphere, gas exchange occurs at the interface, at the surface of the ocean. This is right at the interface between sky and water. So the upper layers of the ocean are much more oxygen-rich than deeper water. You have currents and things like this that mix the water so that this oxygen can sweep across the ocean floor and deeper areas and sustain the animal life that lives there. But without these currents, without this mixing, that wouldn't be feasible, and life on the ocean floor wouldn't be as feasible. At least aerobic life. Now, other factors that affect the ability of oxygen to dissolve in water include the temperature of the water or of the solution, where gases will tend to dissolve less efficiently in warmer solutions. This means that warmer water actually holds less oxygen than colder water. So fish in colder water can breathe easier than fish in warmer water, simply because there's more dissolved oxygen available. This is a little counterintuitive because we might be inclined to think that warmer water would be more habitable, more comfortable for the fish, because warm air is comfortable for us, but it's actually not the same. Colder water has more oxygen and can, in general, be more comfortable for many species of fish. Now, the concentration of solutes within the solution can also influence gas absorption. In this context, more solutes generally means that less gas can be absorbed. Seawater is rich in solutes, and it's thus much less absorptive of oxygen gas than fresh water, like you'd find in lakes and rivers. Also, rivers that are fast-moving and rugged, with lots of rapids or splashing areas, will have, these are areas of relatively high surface area to volume ratio. And this relatively high surface area of the water, or I should say the interface between water and air, this will improve gas exchange, and it increases the oxygen content in the water. Lastly, and perhaps most importantly, Gas absorption in a liquid solution will also follow the law of partial pressure gradients. So if there's a higher partial pressure of gas in a water solution than there is in the atmosphere, the gas will diffuse out of the water and back into the atmosphere. And this also works in reverse. The ambient biology of a marine habitat, for example, can also influence the gas environment. If there are photosynthetic organisms in the water, for example, like cyanobacteria, their metabolism in the presence of sunlight can produce a lot of oxygen that can diffuse into and enrich the water body that they happen to live in, like the ocean along the coast, or in a river or a lake or what have you. Conversely, clouds of zooplankton and other microscopic organisms can blossom in the presence of all of this oxygen and consume it and eat it away. And then when they've run out of oxygen, they'll all die, and their decomposing bodies will use up any other oxygen, and they can really quickly create these anoxic zones in the water body. These are all really important dynamics of gas movement that you should at least be aware of or familiar with before I go into any more detail here. So to really dig into the biological animal aspect of gas exchange, we should first look at the different structures that animals have evolved to respire, like gills and tracheal networks and lungs. Now, the gills are present in marine animals, as gills function to pull oxygen out of the surrounding water. In the invertebrates, the gills are widely varied. They can be internal, running along the inside wall of some body cavity and requiring cilia or a limb or something to move water over them, or they can be external, like a tube worm with a flashy hair-like protrusion of thin cylindrical gills. The mollusks, for example, possess just 1 or 2 gills, which take the shape of feathers or feathery tufts of tissue. If you like looking at pictures of marine animals, chances are pretty good that you've probably seen images of some kind of aquatic gastropod that has 2 feather-like things protruding from its head. These are its gills. Now, in many mollusks, the gills are used for more than just respiration. These feather-like structures are also used like sweeping limbs or flippers that can help to direct water flow inside the mollusk's mantle cavity. The gills can also be used to close off openings to the mantle cavity by obstructing water flow, so as to keep sediment or pollutants out. The sponges are perhaps the most primitive animals of all. They don't have gills, they don't have tracheal networks, they don't even have lungs. The sponges really are just lumps of biomass that have a network of pores and channels running through their bodies, and they get everything they need from the water that flows through this pore network. The same features, the same anatomical structure of the sponge, absorbs nutrients just as it absorbs oxygen and releases CO2 waste. The echinoderms are an ancient invertebrate animal lineage, but they're definitely not as primitive as the sponges. However, despite this, rather surprisingly, the echinoderm respiratory system is remarkably simple and rudimentary, having been integrated into their greater water vascular system. The echinoderms use this internal pressurized network of water-filled columns and veins and cavities to move their tissues. If you take a starfish, for example, lift it up and look at the underside of it, you'll see that it has all of these short, stubby little tentacle-like things coming off of the entire bottom surface. These are pressurized, water-filled tubes, and they function like little mini suction cups or like suction columns. And by using this pressurized water vascular system, the echinoderms can move these little suction cups and grasp on to new areas in their environment to slowly wiggle their bodies over. This is how they move. And it's also partly how they perform respiration, because along the sides of their body, some of these water-filled pouches of the water vascular system, they bulge outwards. They form these sac-like protrusions coming out of the side of the echinoderm. These are called diverticula. And now some of these bulging protrusions, these diverticula, the flesh is stretched out and it's pretty thin. And so they're used as a local source of oxygen and CO2 exchange, although the whole efficiency of this system is relatively poor. Aquatic arthropods, including many species of insect, have evolved gills or gill-like respiratory structures on their limbs. Or it actually might be more accurate to say that they've evolved to repurpose some of their limbs or limb segments to be used as gills. And these gills are often the interface between a sealed inner tracheal network and the watery external world. And of course, I'll get into the tracheal network in much more detail in a few minutes. But now, let's compare these invertebrate gills to the vertebrate gills. Vertebrate gills, like you'd see in fish, are much less varied. There's really only one basic design. The gills exist on either side of the head, immediately behind the mouth, and they include 4 arches, or columns of tissue, lined with gill filaments. The gill filaments are covered in even smaller structures called gill lamellae, which are like thin nodes or sheets that take the form of like deep ripples. Now, whatever structure they take, these gill filaments are designed so that water flows over them in one direction. The gill lamellae are aligned such that water will flow between them, which helps to maximize surface area. And the lamellae are themselves packed with capillary beds that have a massive surface area that can easily absorb all of this incoming oxygen out of the water. The capillary beds themselves are part of a circulatory network organized into a counter-current arrangement. Countercurrents are 2 currents that are parallel but flowing in opposite directions, and in biological systems, that can have some really important advantages. Namely, countercurrents allow for the maintenance of the partial pressure of oxygen and carbon dioxide across the entire gill, and this allows for steady, continual oxygen absorption, even as the concentration changes over space. So this is what I mean by that. The water will flow in one direction over the gills, from front of the gill to the back. And as the water flows over the gill in this one direction, the oxygen in the water will get absorbed into the animal's tissue. Now, the water at the front of the gill, the water that initially makes contact with the gill tissue, this water has the normal oxygen content. It's relatively high in oxygen. But as it flows over the gill and oxygen is extracted, by the time the water gets to the end of the gill, it's had as much as 85% of the oxygen removed. So the water flowing over the end of the gill only has 15% as much oxygen. Now, the blood in the animal's tissue is flowing in the opposite direction. The blood is directed through the circulatory system to come from the body, where it runs along the backside of the lamellae from the rear of the gill, and then it flows through the capillary beds to the front of the gill. At this point, it runs out of the gill structure and returns to the body. So the blood and the water both share the same pattern of oxygen concentration, rich at the front and poor at the back. At every point along the length of the gill, the oxygen's partial pressure inside the animal is lower than it is at the point immediately outside the animal. So oxygen is always diffusing in, even though the concentration both inside and outside is changing across space. This makes gills remarkably efficient and thorough respiratory organs, and the marine animals need them. They need them to be this thorough and efficient when they're in a watery environment where they may not have an ideal amount of oxygen. However, a problem is that because of their surface area and their high vascularity, gills are also a place for water and solute exchange, which is usually a complication for the marine animal. Now, I talked about this in the episode on animal hydration, but to briefly summarize that content so you get a little refresher, saltwater fish can lose water out of their gills, and freshwater fish can lose solutes out of their gills. And both of them have evolved respective mechanisms of osmoregulation to restrict that outward flow and keep themselves in balance. But understand that this is largely an aquatic problem. Terrestrial animals don't have to worry that much about solutes, like nutrients, leaking out of their mouths, because air simply doesn't facilitate that kind of diffusion like water does. Well, I should say, to a point. You have all kinds of vertebrates and invertebrates that live on dry land and that breathe dry air, and they've all had to evolve different systems to extract their oxygen. Now, the biggest threat for these organisms is dehydration through water evaporation, but this is largely a problem for smaller animals, like insects, where just a tiny change in the internal water concentration can be quite damaging, and that change in concentration can happen relatively quickly because they're so small. Now, larger animals, like many mammals, for example, will pant or sweat so as to exploit the phenomenon of evaporation to cool themselves down. So it's not a risk for them, it's actually a built-in mechanism. Water loss through regular breathing, it's not a threat to larger animals, but to insects, even regular breathing can be potentially dangerous. So why is this? Well, the insects breathe through an organ system called a trachea, or a trachea network. And this is like a series of vascular tubes that run throughout the insect's body, and which reach out to the surface at various points to create spiracles. These spiracles are pores leading into the tracheal network, and they can be opened or closed at will. The tracheal network itself is literally an open series of passageways and corridors that permeate the insect's body, with highly branching tips that innervate as much of the insect's body as possible. Now, in very, very tiny insects, the size of their bodies allows oxygen gas to flow into and through their tracheal network, and supply all of their tissues with oxygen, all through simple diffusion alone. This unaided form of gas-based circulation is only possible because the distances involved are extremely tiny, because the insects are very small. Larger insects can't rely on diffusion alone. This is a mechanism that doesn't work over larger distances. And so these larger insects had to evolve a few more tricks that enabled them to evolve to be so large. First, larger insects have disproportionately larger tracheal tubes. This is to say that if you took a small insect and somehow enlarged it while keeping its proportions, it would have smaller tracheal tubes than an insect which is naturally that larger size. The evolutionary purpose of these larger tracheal tubes is to exploit the cross-sectional area component of Fick's law and enable greater gas flow throughout the insect's body. Furthermore, insects have muscle tissue that they use to flex their bodies, to move their legs, to flap their wings, and to wave around their antennae and shake their abdomen. The flexion of all of these muscles, especially the larger muscles used to walk or fly or perform signal movements with the abdomen, this flexion can create distortions and contractions in the tracheal network. And by using their muscles, the insects can change the size of passageways and the overall volume of regions of the tracheal network. This changing volume can change the internal pressure, and this active dynamic flexing of muscles, typically in a rhythmic pattern like legs walking or wings flapping or abdomen vibrating like in bees to send a message to other bees, this can create internal currents and airflow. And this can not only improve the flow rate of gas and thus improve gas exchange within the animal's tissues, but it also allows for the capacity to breathe gases in and out. The size of a large insect is, in part, constrained by the oxygen content of the atmosphere. If there was a higher oxygen content, like that which existed during the Paleozoic era, this could supply a higher partial pressure of oxygen, and thus improve its solubility in solutions. When partial pressure of oxygen was higher, gas exchange was easier, and insects could grow much larger bodies. And back in the Paleozoic era, there were species of insect that grew up more than 2 feet in length. Nowadays, our 21% atmospheric oxygen content and its partial pressure are significantly lower than they were in the Paleozoic era, and so large insects have to balance fitness trade-offs. They can't be so large that they can't diffuse oxygen to all their tissues, but they also can't make their tracheal passages arbitrarily large, because this takes up, internal space, and they can crowd out room inside the exoskeleton for organs and muscles. And the muscles would have to be strong enough to move these arbitrarily large tracheal passages. And as the muscles had to be stronger, they had to be bigger, and so you get all of these competing fitness trade-offs. Bigger passages, bigger muscles, bigger body size. How do you balance it all? Among the insects, this is a really important evolutionary dynamic that all of these different species have tried to solve in different ways. Now, the land-dwelling vertebrates, like the elephants and monitor lizards and the frogs and the moose and gazelles and humans, they all have, in my opinion, the most interesting respiratory structures: the lungs. I should mention that some fish and even some invertebrates also have lungs, but it's a universal trait among the land-dwelling vertebrates. In this respiratory system, oxygen is inhaled through the mouth. It's carried through a part of the throat called the trachea, which splits into the left and right bronchi, feeding into the respective lung, and each bronchi ends up branching and splitting into smaller passages called bronchioles. Now, this is the general core evolutionary structure of this form of respiratory structure. But from this point on, depending on which species you're looking at, these respiratory systems tend to differ between major animal lineages. They've all had their various evolutionary divergences and tweaks and modifications. One major point of divergence is the use of positive or negative pressure ventilation, or to put it another way, how oxygen is brought into the body and how carbon dioxide is expelled. For example, in frogs and other amphibians, they tend to use positive pressure ventilation, where the oxygen that's brought into the mouth is pushed into the lungs instead of sucked into the lungs. A frog will lower the floor of its mouth, increasing the internal volume and thus pulling air in through the nose. Then it will close its nose and use internal muscle contractions to push the air in the mouth down into the lungs. In contrast, mammals will use negative pressure ventilation, which pulls oxygen into the lungs. So this is kind of similar to how the frogs pull oxygen into their mouths, but the frogs have to push it into their lungs. In mammals, in contrast, to inhale, the mammal will lower the diaphragm, which is a muscular structure that exists below the lungs. So this descending, contracting motion of the diaphragm, plus the corresponding expansion of the ribs, will stretch the lungs and increase their internal volume, and this decreases the internal pressure relative to the outside air. Air will then follow this pressure gradient and flow into the lungs. Then, when the diaphragm and the muscles around the ribs are allowed to relax, the lungs will return to their original size, and any excess gas will be forcefully expelled in an exhale. So the frogs and amphibians, they force the gas down into the lungs. But in mammals, they force the gas out. This is the difference between positive or negative pressure ventilation. In the context of strange evolutionary tweaks and divergences to this respiratory system, let's look at the birds, because the birds have really rather strange lungs, where the air flows not in and out along the same tract from inhalation and exhalation, but rather it flows in a single direction. When the bird inhales, air flows through the trachea into two large air sacs that are held behind the lungs. When the bird exhales, this air in these air sacs is pushed from the sacs into the tiny bronchi of the true lungs. Now the bird can inhale again, and more air is brought into these just emptied posterior air sacs. The air that's currently in the lungs from that first inhalation gets pushed up to the front of the lungs, where it then flows into a series of anterior air sacs. So these anterior air sacs hold the air from that first inhalation. The bird has that second inhalation, which has moved gas back to those posterior air sacs, and then the bird now has its second exhale, where the air in the anterior air sacs, the ones up front, is expelled from the body. So the first air initially inhaled, now it's being finally removed from the body after this second exhale. And the air that's in those posterior air sacs then moves up into the lungs. So this is a pretty complicated system, but it's powerful because it enables unidirectional airflow. And when you're flying through the air, this efficiency bonus is incredible. Birds had to evolve a tremendously efficient respiratory system because flying is really hard. It takes a huge amount of energy to fly. And two, when you're flying really high up in the air, the atmospheric density is much lower, so there's a lot less available oxygen. This unidirectional respiratory system allows birds to use air more efficiently, as they can absorb oxygen during both inhalation and exhalation, as opposed to absorbing it during just exhalation. Also, the blood vessels run perpendicularly along the bronchi in the bird's lung, making it more efficient at extracting oxygen out of the air than the alveoli of a mammal. But it's still less efficient than the gills of a fish. Now, animals of all types have to regulate their rate of respiration so as to keep an internal balance of the oxygen and carbon dioxide in their tissues. If an animal were to hyperventilate, for example, they're not getting enough oxygen in and they're not getting enough CO2 out. And so what happens is they start to get a buildup of CO2 in their tissues, and this can be pretty harmful. In another example, during vigorous exercise, tissues will use more oxygen because they're engaging in more activity, they're producing more ATP, and they'll also produce more waste carbon dioxide, so the body's respiratory needs are all increased. This also, respectively, decreases and increases the partial pressure of these gases in the blood. The CO2 in the blood can react with water to form carbonic acid, which then dissociates to form a proton and a bicarbonate ion. So the end result of this is that when you exercise, when you engage in physical activity, your cellular activity is going up, you're using more oxygen, the end result is that there's more CO2 in the blood in the form of bicarbonate ions and free protons. And this can make the blood slightly more acidic. And this can potentially be dangerous because acid signals can kill cells. Now, the blood itself is a connective tissue composed of cells within a fluid matrix. And these cells include platelets, which are small cell fragments that physically and chemically promote clotting, which is, of course, the first step in wound healing. Then there's white blood cells, which are immune cells that help fight off pathogens. And there's the classic red blood cells, with their iron-containing hemoglobin proteins that bind to and carry around oxygen and protons. Hemoglobin is a critically important protein because it increased the oxygen-carrying capacity of the blood, which could then in turn support a more cellular respiration, more energy production, which then directly supports the growth of larger tissues and larger organisms. So hemoglobin, in other words, is really important. It's what allowed larger animals, more complex animals, to evolve. Now for a little bit of chemistry. If we look up close, we see that a hemoglobin molecule is a tetramer, which means that it's composed of 4 smaller protein subunits. Each one of these subunits is a protein that wraps around a non-protein molecule. It's an iron-containing molecule called heme. So a single hemoglobin molecule has 4 of these proteins, each with a heme molecule. So every hemoglobin molecule has 4 iron molecules. The oxygen can bind to the iron ion in the heme, which means that each hemoglobin molecule can carry up to 4 oxygen atoms at a time. What's really interesting is that hemoglobin exhibits cooperative binding, where the binding of one oxygen atom will change the conformation of the other subunits, so that they become more likely to bind to another oxygen atom. So you bind one, and all of a sudden the hemoglobin has a higher affinity for oxygen, so it's more likely to bind another. And that binding will change the conformation again to increase the affinity stepwise again. And so this happens until the hemoglobin rapidly picks up 4 oxygen atoms. Conversely, as oxygen gets removed, the conformation will change back. And in a stepwise fashion, this can reduce the affinity for oxygen and make hemoglobin more and more likely to release the oxygen it's carrying. This exceptional quality of hemoglobin makes it really good at either picking up a full load or dropping off a full load of oxygen. And this is why it's so good at shuttling around oxygen throughout the body. Whether the hemoglobin is actually picking up or dropping off oxygen, that depends on the partial pressure of oxygen in the blood at that cellular site, at that tissue region. And hemoglobin happens to be particularly sensitive to the partial pressure of oxygen. For example, during exercise, the partial pressure of oxygen in the blood will drop as muscle cells consume the oxygen at a high rate. This relatively small change in the partial pressure, like a change of 10 millimeters of mercury, this can lead to hemoglobin dumping 25% more of its oxygen into cellular tissues. So the hemoglobin is stepping up the oxygen supply when it senses low oxygen concentration. Earlier in the episode, I talked about the qualities of a liquid solution that can influence gas diffusion, such as temperature and solute concentration. Well, the blood is a liquid solution, and its temperature and pH can influence the activity of hemoglobin. For example, more CO2 in the blood makes the blood more acidic, as the CO2 reacts with water to form carbonic acid, and the subsequently lower pH makes hemoglobin actually more likely to release its oxygen. Now, this is a neat evolutionary tactic, because it means that less CO2 will increase the pH of the blood and make hemoglobin more likely to pick up oxygen, which is exactly what you want in a place like the lungs, where most of the CO2 has been released. The blood pH is relatively high, and there's incoming oxygen ready to be picked up and distributed to the tissues. Another interesting aspect of the circulatory system is the pH regulatory strategies that are used to help buffer against the CO2 concentration. So when CO2 is released by the tissues as a product of their metabolism, the CO2 will get absorbed by red blood cells, where it reacts with water in the cytoplasm and the enzyme carbonic anhydrase, and this produces the carbonic acid. And then the carbonic acid will decay into bicarbonate ions, HCO3-, and protons. Now this conversion will consume the CO2, and it lowers its partial pressure in the blood cell, which then encourages the blood cell to absorb more CO2 out of the tissues. The subsequently produced bicarbonate ions are expelled out of the blood cell into the blood plasma. And so what this means is that most CO2 actually travels in the blood through the plasma in the form of these bicarbonate ions. The CO2 isn't actually carried by a red blood cell. Now, the protons from this carbonic acid breakdown are contained within the blood cells. The protons are attracted to hemoglobin with a few oxygen atoms attached, and so this effectively soaks up the protons and removes them from the blood, which helps to decrease the blood's acidity. So in other words, this means that hemoglobin, especially when it's loaded up with oxygen, is a buffer that helps to control blood pH. So when the hemoglobin is at the lung environment and it picks up oxygen and it picks up these protons, that'll help reduce the acidity that you can see at the lungs. So it helps to regulate. It's a top-down regulator. And you'll see that a lot of biological systems have features like this, competing mechanisms or competing influences that keep some variable in a safe range or in check somehow. Okay, so I'm clearly starting to talk more and more about the circulatory system. So I want to briefly explain a few more details before tying it all together. The circulatory system is the complex series of veins and arteries and countless capillaries that the blood flows through. Circulatory systems are typically found in larger animals, where this kind of vascular network is the only reasonable way to sustain their size. It's the only reasonable way to distribute nutrients and oxygen throughout their large bodies. Diffusion doesn't cut it. Smaller animals, as I've said, they can rely simply on diffusion to move oxygen and stuff through their bodies. When we're talking about circulatory systems, there's two general types, open and closed. Now, an open circulatory system is often found in insects, where the heart will move a fluid called hemolymph around the body. This circulatory system is considered open because the hemolymph isn't exclusively confined to blood vessels. The oxygen is simply mixed around the hemolymph like a ceiling fan mixes air in a room. Nutrients and oxygen don't necessarily have to diffuse across the wall of a blood vessel, as the hemolymph is often in direct contact with the insect's organs and muscles and other tissues. So this is a low-pressure system that does not efficiently move around oxygen. It's common in simpler, more sedentary animals with low oxygen demands. Many insects, however, are highly active and energetic, and they compensate for their open circulatory system with their tracheal network. The tracheal network complements this open circulatory system and compensates for its inefficiencies. Now, the closed circulatory systems, on the other hand, are entirely confined within blood vessels. The blood flows through the vasculature in a circuit, running back and forth from lungs to tissues. And thanks to their structure, closed circulatory systems have much higher pressure and much faster rates of flow than open circulatory systems. Furthermore, blood can be preferentially directed to tissues with higher oxygen demands or with a lot of waste CO2 production. The blood is propelled via the pumping of the heart, which is a powerful muscular organ that rhythmically contracts to create pulses of high pressure, propelling the blood fluid, the blood matrix, through this circulatory system. Now, in all animals, the heart serves the same general function, but it may appear slightly differently, or the details of its function may be slightly different. So for example, the heart has a different number of internal chambers, depending on the species. All animals have at least one atria, which is a chamber where blood flows into the heart, and they all have at least one ventricle, where the blood gets pushed out. Now, the number of atria and the number of ventricles has increased as animals diverged and diversified. The earliest vertebrate animals, like the fish, have two-chambered hearts, with one ventricle and one atria. But in the slightly more complex amphibians and many reptiles, which have adapted to life on land, drier conditions, they have three-chambered hearts, with two atria and one ventricle. Now, the mammals, the birds, and the crocodilian reptiles all have 4-chambered hearts, with 2 ventricles and 2 atria. This increasing complexity developed as a response to the increased difficulty of pushing blood in a terrestrial body, where there's no water for buoyancy, and the force of gravity is a lot more noticeable. The heart has to be stronger because it has to pump blood with more pressure, but the veins and the arteries are pretty delicate, and so there's a balance that has to be struck here. There's a fitness trade-off. You pump blood too hard, the veins and arteries and capillary beds will rupture, and you can have internal bleeding and die. So that's bad, and evolution generally wanted to avoid that. It cleaned up the animal permutations that had that flaw. And eventually, these terrestrial animals evolved separate circuits for their blood flow. They have the pulmonary circuit, which is a relatively low-pressure circuit that carries blood a short distance between the heart and the lungs. And then there's the systemic circuit, which is relatively high pressure and carries blood between the heart and the rest of the tissues and the organs in the body. So in the fish, the circulatory system is pretty simple. The heart will pump blood to the gills, where it picks up oxygen and flows to the body. The body tissues will consume this oxygen, and blood will flow back through the heart to the gills again to get reoxygenated. Now, in the amphibians and many reptiles with a 3-chambered heart, the ventricle pumps out oxygenated blood from the lungs, deoxygenated blood from the body, and mixed blood. Increasing cardiovascular sophistication has minimized this mixing, so as to optimize the available oxygen per heartbeat. In the crocodilians, for example, this mixing of the oxygenated and deoxygenated blood still exists to some degree, even though they have a 4-chambered heart. In the birds and the mammals, this mixing of the 2 blood types has largely been phased out completely. In the birds and mammals, the deoxygenated blood flows from the body through the venules and the veins into the right atrium of the heart, which pumps it into the right ventricle, which then pumps the blood into the pulmonary artery. Here, in the pulmonary artery, the deoxygenated CO2-rich blood will go to the lungs to pick up oxygen. The partial pressure gradient in the lungs will encourage the CO2 to diffuse out of the blood through the alveoli and out of the body. And this is due in part because the partial pressure gradient will cause protons to detach from the hemoglobin and react with the bicarbonate ions to form CO2, which will then diffuse out and be exhaled. And as the CO2 is being removed, the pH of the blood increases. So you have not only the CO2, but also these free protons that are all, binding together and then leaving. And taking those protons away is what reduces the acidity and increases the pH. This increase in blood pH makes the hemoglobin more likely to pick up oxygen. After the CO2 is exhaled, an inhale will follow, bringing in the oxygen that can then bind to the hemoglobin. Once the blood has been oxygenated at the lungs, it'll return to the heart by flowing through the pulmonary vein into the left atria. Then this oxygenated blood is moved into the left ventricle, which is really strong and powerful because it has to pump really hard to provide the force to pump this blood through the whole body. The left ventricle will pump the blood first into the aorta, which is a huge, similarly thick blood vessel. From the aorta, the oxygenated blood is distributed throughout the body. In the capillaries, as the blood is flowing through individual cells, these cells will tap the hemoglobin for oxygen. In return, they'll release their waste CO2. So even though the heart is just one organ, in these more complex animals, the heart is simultaneously pumping blood through 2 distinct circuits within the body's entire circulatory system. And the combined effort is a perpetual flow of blood throughout the animal's entire body that enables not just the distribution of nutrients, but the distribution of life-giving oxygen and the removal of carbon dioxide waste. And that, ladies and gentlemen, is how animals engage in gas exchange. I hope you enjoyed this episode, because the chemical efficiency, the physiological speed of these systems, and the evolutionary importance of gas exchange and circulation are they're so amazingly fascinating and just a wonder to research. So I hope for you it was really enjoyable to listen to and to learn about. Now if you like this episode, then hit that like button. And if you like all my content and you want to see more, then hit that subscribe button and hit that bell too, because YouTube can be kind of iffy about this, so that whenever I post something new, you can see it right away. And if you're enjoying this series, part 2 on animal physiology, then you should also hit that subscribe button so you can see the rest of these episodes when they get posted. Stick around for the next episode on the animal immune system. So if that sounds fascinating, if you want to learn more about that, then subscribe. And as always, thanks for listening.